Let's come back to our main topic, viruses. And now we need to discuss one unique type of virus. We said already that viruses actually can infect wide range of alive beings. We have viruses of plants, of animals, viruses of birds, insects, and even we have viruses which can infect bacteria. And virus that infects bacteria is known as bacteriophage. So origin of the word is similar to our immune cells. Phagein in Greek it is to devour, and bacteria, of course, it is bacteria. So literally, bacteriophages are virus which can eat bacteria. And of course, we have macrophage, we have microphages, means cell which can eat something. Of course, bacteriophage cannot directly eat bacteria, but that virus can destroy bacteria. And initially, it was named bacteriophage. That name is still used, and uh, actually, we again identify any virus which can infect bacteria as bacteriophage. So as well, we have bacteriophages which can affect even archaea, but of course they not of medical significance. And mainly we'll talk about viruses which are dangerous for bacteria. So viruses of bacteria are very similar by properties to animal and human viruses. They have more complex structure. We'll talk a little later, but as well, bacteriophages have unique receptors or structures which can recognize only one unique type of bacteria. And actually, even that species we usually use in our medical language, it's not really important for viruses because, again, species is some bacteria, some group of bacteria which have common properties such as biochemical, morphological, and tinctorial. But of course, bacteriophage will not care about biochemical property or some other properties. It is a virus as any virus, it needs a receptor. And structure of bacteria is organized so that species, it is something identified by human. If we see that bacteria have common properties, we say that it is species. But viruses will recognize only one unique receptor. And it occurs so that even species of bacteria can be very diverse by structure of those receptors. And in microbiology, we have such a group of bacteria which is called phagotype. Phagotype of bacteria, it is a subgroup of bacterial species susceptible to a particular bacteriophage. So it is always some smaller than species subgroup of bacteria which is susceptible to the same particular bacteriophage. And of course, to identify that group, we need to use method which is named phage typing or phagotyping. It means that you will take different bacteriophages and inoculate with bacteria. And if bacteria will be affected, some group of bacteria will be affected by the same phage. It is phagotype. It is one phagotype. And one species of bacteria of every bacteria can be subclassified or subdivided into multiple phagotypes. So again, please don't be confused if you will be asked about phagotype of bacteria. It's not something related to macrophages, even if word is very similar. It's not a group of bacteria which can be eaten by the same macrophage. Macrophages don't care about structure of receptor. They care about antigenic presentation. They will engulf any foreign particle. But phagotype, it is a group of bacteria affected by the same bacteriophage, and phage originates from bacteriophage. That's why phagotype. So later we will show how to use it, but first of all, of course, we need to describe structure of bacteriophage. And again, it is a virus, and common principle is the same. It should have genetic material which can be presented by both DNA and RNA, and some accompanied structures. So bacteriophages can be very diverse in structure, and we have the classic example of most complete virus is a T4 phage. It is shown in current slide. that virus have the maximum possible number of structures which can be identified in bacteriophages arrow. So all possible structures are present. It can be found in T4 phage. It is again DNA in the case of T4 phage, but actually bacteriophages can contain RNA as well. It is a capsid, which in the case of bacteriophage called as a head. And if you take a look on that picture, that phage is looking like a bug, like some insect. And actually name of different structures are given in corresponding manner. So capsid is called as a head. It is connected with the body of bacteriophage through neck and maybe collar, and that elongated body can be as well named as the tail of the phage or sheath of the phage, and it's ended with the help of base plate and pods or organelles which are required for attachment to the surface. Pods can be as well identified as the tail fibers. And between those fibers, T4 phage have as well teeth or pins which are required for better attachment to the surface or to make the hole in the host cell. So as far as bacteriophage is a virus infecting bacteria, the host for bacteriophage is bacterial cell. It's not animal or human, it is bacteria, means prokaryotic cell. And actually, viruses are the only microorganisms which can affect everything. Any life organism can be affected by viruses. We have viruses of everything, and actually we have even virus of virus. If you talk about some hepatitis B and D, hepatitis D is kind of parasite of hepatitis B, so we can say that viruses can infect even viruses. And bacteriophages are viruses of bacteria, so they can recognize bacteria, stick to it, and infect bacterial cell. That is, as we said already, the most complete type of bacteriophage, but we have multiple different families of bacteriophages, and they as well classified by common principle of viruses. They are classified into DNA and RNA containing. They can be single-stranded or double-stranded. They can be naked or enveloped, and they can have different number and different combinations of organelles, as you can see on that picture. So you will not be asked about classification according to family and other. It's not really important in the case of bacteriophages, but just for you to know and to imagine how many actually different bacteriophages we have, that picture is provided. So again, if you talk about type of replication of virus, it is very similar to human viruses, almost the same steps. But in the case of bacteriophages, we have one unique phenomenon. And according to that phenomenon, bacteriophages are divided into two major groups, lytic bacteriophages and lysogenic bacteriophages. So lytic bacteriophages, as well known as virulent phages, they take over the machinery of the cell to make phage components. So the main principle is the same as for viruses. And after viral reproduction, lysis of the cell will occur, releasing of new phages particles will occur, and cell, as the result, will die. So in life cycle of lytic bacteriophages, we have several steps of their multiplication. They are again similar to our human viruses. First step is absorption. Virus need to recognize suitable receptor. Again, it is very specific to only small group of bacteria, even smaller than a species of bacteria. And virus will attach to the surface of bacterial cell. After that, genetic material will be injected in the cell. Usually that tail is required for injection. It is elongated in complete particle, but then some amount of energy is present in the tail, and it have contractile teeth or some fibrilla. It will contract and genetic material will be injected in the cell, and the whole coverage of the phage will remain on the surface of bacteria. And if you will perform electron microscopy of that bacteria, you can identify capsid or head and tail of the phage on the surface. It will stick here, it will remain on bacteria, and it is very often named as a ghost phage because it's looking like some silver symbol of a ghost, and bacteria will live with that thing until it will break. So in the next stage, DNA or RNA of bacteriophage will be incorporated in the life process of bacteria, and similar to other viruses, different parts of bacteriophage will be synthesized in different sites of bacterial cell. Proteins in one, enzymes in another, genetic material in the third part, and during that stage, again, organelles or compounds of virus will accumulate in the cytoplasm until cell will not be exhausted. When all energy or some proteins or other compounds will be exhausted, bacteriophage will start assembly of virus, means all particles will be delivered into the one site of cytoplasm, and they will be arranged in complete viral particles. And after that, again, those viruses will make holes in cell, lysis of cells will occur, and new viral particles will be delivered into the environment, and they can infect new bacterial cells or spread by some mechanisms to other locuses, to other sites. So that is the complete lytic cycle of bacteriophage. It is very simple, and again, it resembles common viral infection. But besides lytic cycle, we have lysogenic cycle or lysogenic phages, as well known as temperate phages. The main difference that after infection of the cell, two first steps will be identical: absorption and entry. But after that, genome of virus will be incorporated to main chromosome or nucleic acid of bacteria of the host cell, and virus will be inactivated. It's just present here in absolutely inactive stage. So that part of viral chromosome integrated into the bacterial chromosome, part of viral genome incorporated into bacterial chromosome is known as a prophage. It is the third stage, and as well, that part of genome is called prophage. It is bacteriophage genome integrated into the host genome. And bacteria will exist with that combined genome. And while bacteria is multiplying, every new bacterial cell will occur genome of that bacteriophage. And actually, bacteriophages multiply not by destruction of the cell. It will accumulate its genome in the population of bacteria. And every new bacteria will have genome of the phage. And that phenomenon, when phage is coexist with normally developing bacteria, when genome of phage is integrated into the genome of bacteria and multiplies with it, is known as lysogeny. So lysogeny, it is a phenomenon of coexistence of phage and normally developing bacteria. When genome of phage is integrated into the genome of the bacteria and multiplies with it, means bacteria remains alive, intact, it's multiplying, and phage multiplying together with bacteria. And in some time point, some factor can act on that bacterial cell, and one of that bacteria, or maybe several bacteria, will be transformed to lytic cycle of bacteriophage. Genome of bacteriophage will be disintegrated. It will go into the cytoplasm of the cell and activate mechanism of viral synthesis, assembly, and release. And lysogenic cycle for one precise bacteria will be ended, and it will be transformed to lytic again. So that's how bacteriophage can multiply its number without any harm to bacteria, but then again it can disintegrate and go into environment to infect newly cells. So why it is important to know, and why we have that phenomenon marked as very unique and very specific. So we have a method of gene transfer in bacteria from donor to recipient with the help of bacteriophages. It is known as transduction. So transduction, it is a method when gene transfer into bacteria from donor to recipient with the help of bacteriophage. So in that phenomenon, bacteriophage first need to infect donor bacteria, and usually when genome of phage will be disintegrated from host cell, only genome of phage should be cut and removed. But of course, viruses are very primitive beings, and they have no mechanisms of control of enzymes, and it is highly possible that bacteriophage will accidentally grab some genes from bacteria, from donor bacteria. And when that bacteria will infect new one, genes of microbe will be incorporated in the genome of bacteria together with the phage. And in that case, bacteria will acquire new property. For example, it is very perfect, and one of the most classic examples, Corynebacterium diphtheria. It is microorganism which is causing serious disease in human known as diphtheria. And the disease is related to production of exotoxin, which is called diphtheria exotoxin. And the toxin initially not belong to bacteria. It is a gene which some years, some centuries ago were acquired by bacteriophage. And now we have bacteriophage which can infect Corynebacterium and integrate gene coding unique toxin. And naturally, Corynebacterium diphtheria have no toxigenic properties. It is not dangerous for human. It cannot cause a disease. But if that Corynebacterium will be infected with tox phage, means bacteriophage will have gene coding for that toxin. Bacteria became dangerous. That phenomenon is known as phage conversion, when a change in the properties of bacterial cell occurs as the result of infection with a temperate bacteriophage. Bacteria will not die, it will coexist with the genome. And of course, as far as the gene belongs to bacteria, bacteria can use it. It will use it for its purposes, and it can be different property coded by those genes. But if it is acquired through phage, temperate phage or lysogenic phage, it is phage conversion. And while bacteriophage is present in the structure of that bacteria, bacteria will have that property. If bacteriophage will be disintegrated, then bacteria will lose its property. So that phenomenon again is important in medical practice because some bacteria can acquire pathogenic properties due to bacteriophage. And that phenomenon is used in some paramedical science, such as, for example, genetic engineering, because bacteriophages can be used as a vector to incorporate genes in the genome of bacteria. And here we need to say that we have two types of transduction. It is generalized transduction, when all fragments of donor DNA from any region of chromosome have a chance to enter into bacteriophage, and means we cannot predict, we cannot know in which part of the chromosome the gene will be integrated. And usually those bacteriophages undergoes lytic cycle. And we have specialized transduction. In specialized transduction, bacteriophage transfer only few genes, but few restricted genes. And only few fragments of DNA will be integrated. And usually they're integrated in the known locus of DNA. And specialized transduction is carried only by temperate phages. And bacteria, of course, will undergo lysogenic cycle. And that specialized transduction is important because, first of all, pathogenic properties are mainly transmitted by specialized transduction, And in genetic engineering, we can use that phenomenon because we can add required gene to bacteriophage. Bacteriophage will integrate it in the genome of bacteria, and bacteria can produce some property which is important for us. Again, classic example in any book, in your book, in internet, anywhere. It is an E. coli, bacteria Escherichia coli, which is a natural inhabitant of our GIT, which can produce insulin. And of course, you all know that insulin is a drug which regulates blood sugar, and it is required to maintain a normal level of glucose. And we have diabetic patients who have impaired production of insulin. They need to compensate it by injections by parenteral way. And of course, manufacturing of insulin is very difficult. It is expensive. It is complex substance. It is very expensive to synthesize it artificially. And initially, it was obtained by extraction of organs from animals. Of course, again, it is some ethic problems because animals should be killed because of some dose of insulin, because some men want to eat candy or something else. And that insulin is not very good quality. It's difficult to purify it correctly. And usually a lot of adverse events, allergic adverse events, are observed with the help of those insulins. So nowadays we have very simple method. We can take gene coded insulin, introduce into bacteriophage, and with the help of specialized transduction, that phage will integrate gene in E. coli. And E. coli will use the gene and produce insulin, thinking that it is some useful enzyme. And that insulin can be easily filtrated and used in medical practice. So it's cheap, it's human insulin, it's already purified, and of course it's highly available. So that phenomenon of transduction is widely used in genetic engineering. Again, the question about how to cultivate bacteriophage is very simple to answer. If bacteriophage is a virus of bacteria, then to grow bacteriophage, you need to inoculate it in culture containing bacteria. Of course, it will not grow on agar without bacteria. But if you inoculate bacteria on agar and then add virus, virus will grow. And actually, the most popular method of bacteriophage cultivation is a plague method, when bacteria required virus and agar are mixed together. They slightly heated. Agar can be easily melted. It became liquid. You need to mix all those components and cool down. Then you will have a petri dish, as it is shown in the slide, with agar, bacteria in it, and bacteriophage. Of course, you need to incubate it in appropriate conditions, temperature and moisture. And bacteria on nutrient medium will start to grow. And of course, if bacteriophage can affect that bacteria, it will infect bacterial cells and multiply. And together with bacteria, you will have growth of macrophage, bacteriophages, sorry. And of course, when bacteria will grow in agar, you will have turbid opacity or turbidity. You will have that, as you can see on the picture, agar is semi-transparent, it is opaque. You can see the bacteria is growing here. And in that agar, you will have a place of transparent area. You can see here multiple small dots. In those places, bacteriophage is growing. Virus is multiplying and it's killing the bacteria which is growing in that position. And of course, if bacteria is absent, agar will appear as transparent and absolutely colorless. And you can see those plaques. It's indicating, it's indicator of viral growth. And of course, if you need to obtain bacteriophage, you need to cut some piece of agar here, melt it and filtrate. It's easy to filtrate bacteria and agar, and virus will pass through filters, and you can easily obtain culture, pure culture of bacteriophage. And of course, similar to bacteria, every plague, each plague is assumed to come from a single viral particle. It is a kind of a colony of one bacteriophage. And titer of virus is given in plague forming units. how many plaques you have, it is a plaque forming unit. And of course, you can count how many bacteriophages you had in initial sample, and you can multiply them the same manner. So, of course, if we have something able to kill the bacteria, why not to use that something for treatment of bacterial infections? And idea initially was very great, and nowadays it's rarely used. It's not so popular as we want it to be. But still, we have phage therapy. It is a part of the therapy when bacteriophages are used to kill bacteria. And of course, you should understand that bacteriophages are more safe than antibiotics. If we will talk about antibiotics, they have multiple side effects and they can affect our normal flora. They can kill bacteria which are naturally found in our body. Bacteriophages have very restricted affinity and they can kill only known bacterial cells. So they are much more safe than antibiotics. And of course, they cannot affect human tissues because they have no receptors to recognize our tissues, and our tissues have no receptor for bacteriophages. So, idea to treat infections was very good, and nowadays we have oral and topical antibacterial applications of phages, which are used even in clinics in human medicine, but mainly in Europe, in Middle East, and Asia. And still, use of bacteriophages is limited and data is not really available. And we know that we have some side effects as occasional inflammation or due to immune response on bacteriophages in that cases. And actually, systemic application of bacteriophages is very limited because we know, we understand that bacteriophage is a virus of bacteria. It's harmless for us. But our immunity cannot understand it. Immune system, immune cells will recognize that bacteriophage is some foreign virus. And of course, if it is foreign virus, our cells need to kill it. And in an attempt to use bacteriophages systemically, till nowadays failed because of very good and strong immune response. We cannot deliver bacteriophages to target organs. They will be inactivated faster than they will get to bacteria. And some companies and a lot of we have some research and attempts to design some particle carriers which can hide bacteriophage of immune response until it will not be delivered to target organ. And only target organ that shell will open and release virus. And virus will have good activity in that studied required organ, required tissue. But still no good carriers are identified, and we have only some studies on animals. And again, they show that most number of phages are taken up by liver and spleen, and bacteriophages are very fast destroyed and activated. They have no therapeutic effect. But still, oral and topical solutions are widely used, especially for treatment of intestinal infections. And nowadays, we have use of inhalational bacteriophages to treat pneumonia, for example, in hospitalized people. It is inhalation solution. You can just breathe with bacteriophage. It will have local activity. It will kill bacteria in lungs. Of course, it will not be absorbed. It will be easily inactivated by even local immune systems. But we don't need that phage to be absorbed. We have activity local in lungs. Bacteria in lungs is destroyed, and we have very good effect. So bacteriophages were kind of forgotten when a lot of good antibiotics appeared. But nowadays, because of antimicrobial resistance and very few number of highly active antibiotics, again, we turning back to the bacteriophages era. And nowadays, some countries already started use of bacteriophages as a standard of therapy of bacterial infections. And maybe in closest future, we will have new therapy options such as bacteriophage. So Again, it is a strain containing bacteriophage for local treatment of some respiratory infections, for example. So we have systemic solutions and we have solutions for local application. But more even popular method of bacteriophage using nowadays, it is phage identification. So we know, and we said already, that bacteriophages are specialized in precise bacteria. And actually, again, the main question first arrived when bacteriophages were identified. Why do we need biochemical activity, multiple biochemical tests, a lot of culture medium, a lot of reactions, a lot of time to identify microbe if we have bacteriophage? And actually, if bacteriophage can kill only E. coli, for example, and you suspect that you have E. coli, inoculate your bacteria, add bacteriophage. If bacteria is not growing, phage killed bacteria, means phage recognize bacteria, and you have a diagnosis. It's much faster and it's more precise. You don't need to evaluate again number of biochemical tests. But again, idea is not very good, and sometimes it is absolutely awful idea, because we have a lot of different phago types for every bacterial species. And the same E. coli, for example, have 50 known phago types. If you talk about some Shigella zone, one species of bacteria have about 64 phagotypes. And if you talk about Salmonella, more than 2,200 phagotypes. And of course, to identify one only group of enterobacteria, you need to perform more than 3,000 tests. Of course, it is absolutely not allowed and not applicable to any laboratory practice. 3,000 tests only for one group of bacteria, it's awful method. And of course, nowadays, identification, routine identification of bacteria with the help of the phages is not performing. But still, we have phage identification. Method is very similar to plague method of phage cultivation. You need to take agar plate, inoculate studied bacteria, split that plate in sectors, and add different bacteriophages against different bacteria. And when a bacteriophage will match the bacteria, it will kill microbe, bacteria will not grow. As you can see on that picture, a petri dish have several circles named with the D1, D2, D3, and everything, everywhere bacteria is growing except sector D5. In D5, you have empty zone, means that bacteria is killed by a virus and agar remain transparent. It is methods of phage identification. And again, you can identify not a species of bacteria, you can identify phagotype. But if you know that that phagotype belongs to E. coli species, for example, of course, we can confirm diagnosis. But usually, we're not using phage identification for primary identification of microorganisms. Usually, it is used to identify phagotype. And phagotype of bacteria, again, it is a group within one species. So why we need phagotyping? Mainly, we have two purposes. First, if you need for example, you have an outbreak of infection, two outbreaks in two different kidney gardens caused, for example, by some Neisseria meningitis, for example. And of course, you need to know, first of all, is it one person who working in two kidney gardens and infecting kids in both facilities? And then you need to find only one subject, isolate, treat, and you will have stop of infection outbreak. Or maybe you have two different persons, and then you need to find two men and to treat both. If you will try to identify a bacteria by routine method, you always will find the same species. They say the meningitis species is always the same. But how to know? Is it the same microbe, or maybe the microbes are different? As one of the options, you can use phagotyping. We know that we have multiple phagotypes within a species, and a chance that you will have the same phagotype in two independent outbreaks is very low. And you need to take samples from one source from one outbreak and from other outbreak and perform phagotyping. If you will see that phagotype is the same, then it is a very high chance that source is the same and you have only one man to treat. If phagotypes are different, then of course sources of infections are different and you have two independent outbreaks. And the second goal, and actually the same purpose we have in some cases when you need to find source of infection. For example, a case of some GIT infection in coffee or restaurant, you need to know who was poisoned or who was infected in restaurant, who not. You need to, again, if you have even the same bacteria, you need to perform phagotyping. If some 10 people will have the same phagotype of bacteria as identified in restaurant, then and it is a high chance that all those people were infected, they ate in the same restaurant. Even if some man was eating in the same restaurant, he or she have similar infection, but phagotype of bacteria is absolutely different, then it is just coincidence that subject became infected at the same time, but actually it's not that case. And second, use of phage typing when you have mixture of bacteria. You have multiple bacteria, it's very difficult to split them, to separate them, but you need to know which microbes are present here. Then again, you need to split Petri dish into sectors and add mixture of bacteriophages. One type of bacteriophage in one sector, one another, one and third, and etc. And sectors where you have growth inhibition, you can see it here, empty zones again, means that bacteriophage recognize bacteria, and you can identify every bacteria present in that mixture of microbes. You will not be required to perform surface plate and isolation of microbes by some mechanical separation, obtaining a colony, and then identify every colony. You can just take phages and identify every species by application of appropriate bacteriophages. So one more, and actually the last modern technique and modern technology which is used with the help of bacteriophages, it is phage display. Technology is very simple by principle, but very important and very useful by the result. So phage display is laboratory technique that uses bacteriophages to find correlation between protein and gene coding its structure. So to be more simple, we know that one gene is responsible for one property, and that property is coded by one unique protein. So we can easily identify gene by identification of start codon and stop codon. And we know that that locus of DNA is one gene. It's easy. But the main task for us is to understand which property is coded by the gene. And to understand property, we need to know which protein is synthesized by that gene. How to do it? Always in cell we have hundreds of different genes, and we cannot know which protein is coded by which gene. So to make it easier, we can cut one known gene or synthesize the same gene, the analog of the gene, and integrate it into the genome of bacteriophage. And for example, that gene, we call it T gene, it is target gene, should be integrated into the known position of genome of bacteriophage. We have, for example, bacteriophage which can insert, perform transduction in known location of bacterial cell. We have always some protein, we call it coat protein, which is located on the surface of the bacteria, for example. We know the structure of that protein. We know which gene is responsible for structure of that protein. So CP gene, it is gene coding for coat protein. And with the help of the gene and bacteriophage, we will insert T gene close to CP gene. And now we have one common gene which is coding for new modified CP protein. And the protein, as the result, will be modified. And new protein will be displayed in that coat protein. So we already know structure of coat protein. We can compare normal protein with new protein, and we will see which protein appeared newly. And that guessed amino sequence can be translated to protein easily with the help of bacteriophages. So phage display is technology, again, it is laboratory technique, which help us to study protein-protein or protein-peptide or DNA-protein interactions. And technique, again, by principle is very simple, but it is extremely important technique because with the help of that phage display, we can translate genetic information of any organism, and we can know which property, which protein is coded by every gene. And we have techniques to translate structure of the gene, but very few techniques how to identify property of that gene, which property is coded by the gene. So actually, that's all for current lecture. Of course, we have a lot. We can talk a lot about viruses, about bacteriophages, and etc. But again, our time is unfortunately limited, and that's all for current lecture. Again, keep your knowledge in your head or in your papers, and don't forget what we said here, because again, in exam and in credit classes, you will have a lot of questions which are described in lectures, but you forgot. So see you next time. Bye bye.